So in the last lecture segment we looked at polyploidy, we looked at aneuploidy, or unusual numbers of chromosomes in cases where you have just a single extra chromosome or a single missing chromosome. We also looked at some of those in Chapter 4 when we looked at some of the sex chromosome aneuploidies, like Turner's female and Klinefelter's male. In this section of the lecture, we're going to look at polyploidy. Polyploids are named based on how many complete sets of chromosomes are in each cell. So you could have a monoploid organism, diploid, triploid, tetraploid, pentaploid, hexaploid, heptaploid, octaploid, etc. Polyploidy is common in plants. In fact, in plants, the more sets of chromosomes, it seems like the bigger, more vigorous the plant is and the bigger the fruits are. And so those are good in terms of agriculture. Polyploid plants are usually the most desirable because you get the most crop yield. You can also find polyploidy in lizards, amphibians, and fish, usually just like triploid. But you won't find humans that are viable, that are polyploid. There are some fetuses that are triploid that have been recovered, but they aborted spontaneously. So you won't have a live person who is a triploid, for example. Now, any plant, primarily we're going to be talking about plants, any plant that has an odd ploidy, an odd number of chromosome sets, will be sterile. And so they won't usually be found out in the wild because they're sterile. Odd ploidies are sterile because if you think about meiosis 1, the first thing that needs to happen is the chromosomes from one set will find their homologs in the other set. And so if you have an odd number of chromosome sets, for example in a triploid, you would have 3 copies of chromosome 1, 3 copies of chromosome 2, 3 copies of chromosome 3, and so on. Then when the homologs pair up, there will always be an extra copy of that chromosome that doesn't have anything to pair with. And that will cause meiosis to stall or to stop or abort, and meiosis won't be able to complete, and so that plant won't be able to make gametes. Or the gametes that it makes will be really messed up. So there's 2 types of polyploidy. One is autopolyploidy, which results from entire chromosome doubling. The entire set of chromosomes doubles in a cell. So the official definition is the addition of 1 or more extra monoploid sets of chromosomes in a cell. And the way that this happens is, in nature, a plant cell can either have a non-disjunction of every chromosome that can give you a whole extra set in a gamete, or in a somatic cell it can happen by what's called an aborted cell division. So we'll look at that. Experimentally, the way they're created is using a chemical called colchicine, which prevents the formation of the spindle apparatus. It causes the cell to be stuck in metaphase, and then if the cell is treated long enough with the chemical, it will eventually abort the cell division and go back to G1 without ever having divided the nucleus. So ultimately, the replicated sister chromatids will detach from each other and act as separate copies of chromosomes. So you'll end up with a doubling of chromosome sets. And I already mentioned that in agriculture, polyploids are desirable because they make bigger flowers, they have bigger roots, bigger fruits. And odd ploidies, because they can't make gametes, will form seedless fruits, which are very desirable, among crops that are like sold in the grocery store, for example. So this is how mitosis, how you can get a polyploid cell in mitosis. you can have replication of the chromosomes, and you can have separation of chromatids, or you can have a cell that's in metaphase and gets stuck, but eventually the chromatids will separate. But if you don't have a cell division that follows that, it's basically what we call an aborted cell division. Then you end up with one cell, not two daughter cells, but just one cell that has double the number of chromosomes as the cell we started with. So this is basically an aborted or incomplete mitosis, where you have the replication part, but you never produce two daughter cells. So that's how you can get a cell that has double the number of chromosomes. So this would be called a tetraploid cell at the end. So for example, if this diploid cell was an embryo, or a zygote even, then if the cell went through one round of this, then you'd have a zygote that had 4 copies of every chromosome. Then you could let that grow up normally and the whole plant would be tetraploid. How you get autopolyploidy because of something happening in meiosis, you can get replication of the chromosomes again. A non-disjunction will produce gametes that are diploid instead of haploid. And then fertilization of a diploid gamete by a normal haploid gamete will give you a triploid zygote, and that's considered a type of polyploid. And by the way, a triploid, if you let this embryo grow up, it will make, if it's a flowering plant, it will make seedless fruits. Okay? And this is just showing you if you had a triploid, or it could be a pentaploid, any odd ploidy, why an odd ploidy cell can't go through meiosis correctly. Because they're showing you how the pairing is going to try to happen. You'll either pair 2 of the 3 homologous chromosomes of each type, or all 3 will try to pair, or there will be no pairing, but it's all screwed up. And you're basically not going to get gametes that have, there's no way to get a gamete that has half the number of chromosomes because you started off with an odd number. So none of the gametes will be normal, and these won't be able to form embryos, which a seed is really a small embryo of a plant. Now what's also interesting in genetics is in plants, in agriculture, is the formation of hybrids. And you can make hybrid plants that have combinations of chromosomes from 2 different species. So when we're looking at these autopolyploidy, what we were looking at was the creation of plants that had multiple sets of chromosomes from one starting set. In other words, just duplications of multiple sets of chromosomes. So you can get a tetraploid plant, but it has the same sets of chromosomes as the diploid plant, it just has twice as many. So the plant type is still the same. It's just a tetraploid version of the same plant. But when you create hybrids is when you cross plants of different species together to make maybe a new species. And that's when things get really interesting. So in this picture, you have a plant, it's a sunflower, and let's say it's diploid. It has 2 copies of A, 2 copies of B, 2 copies of C. The diploid number of the species is 6. It will make gametes that have 1 copy of A, 1 copy of B, 1 copy of C. And then let's say you have another species of sunflower, but its chromosomes are different enough such that you can clearly see a difference here. But it will also make a gamete that has 3 chromosomes, but not the same 3 chromosomes as we saw on the left. In plants, you can get fusion of these 2 gametes. In animals, this is going to be more rare, but in plants, it's a little more common to be able to get fusion of gametes from different species. But what's going to happen is in the initial zygote and then in every cell in the plant, you'll have just 1 copy of each chromosome. Now, I would argue, and I have had this, I have made this comment to Ben Pierce, who wrote your book. I would argue that at this point, this plant is monoploid. It's a new species because it now has 1 set of chromosomes, only. It has one copy of A, B, C, G, H, I, one set containing 6 chromosomes. Now Ben Pierce says this is a diploid hybrid, but I disagree because diploid means 2 sets, and I only see one set of different chromosomes here. All right, but let's assume that this plant, because it only has one set of chromosomes, it doesn't have any homologous pairs, it's not going to be able to make gametes correctly, so non-viable gametes. I agree with that. It won't be able to make viable gametes from this plant. So the only way to make a plant that's viable is to fertilize this gamete here, A, B, and C, fuse it with a gamete that's G, H, and I, and then when that zygote is just one cell big, you force it through an aborted mitosis. Each chromosome will replicate, but the cell won't divide, and that then makes another copy of every chromosome, and that zygote can grow and become a viable plant that can make gametes. And at this point you've created a hybrid plant. It's neither species 1 or species 2 anymore. It's a hybrid of both, and typically you'll get a combination of traits from both, and you'll typically get, because there are more chromosomes, you'll get bigger plants, bigger flowers, more vigorous. So Ben Pierce calls this a tetraploid. I call this a diploid, but it's a diploid of a newly created species. Now one set of chromosomes contains A, B, C, G, H, I, and the second set contains a copy of A, B, C, G, H, I. So you can decide whether you agree with me or not. Over here he seems to say diploid is 12, and over here he seems to say tetraploid is 12. So I think I feel like he's not clear on this or something, but I'm clear on this. We're starting with diploid. This has 2 sets of chromosomes. This is a different species that has 2 different sets of chromosomes. We can hybridize them by fertilizing different gametes together. We create a sterile, the plant is viable, but it can't make any good gametes. So what we would have to do is take a zygote like this, force it through an aborted cell division to create and grow a plant that has 2 sets of everything. And that's how you can create what I agree at this point is the amphidiploid. This is the amphidiploid. It's diploid, and amphi refers to the hybrid nature of it. Now most of your crop plants are polyploids. Some of them are autopolyploids, having multiple sets from the same original plant species. Some are allopolyploids, having combined sets of chromosomes from 2 different species and then being forced through a chromosome duplication event to create fertile hybrids. But the point is that most of your crop plants are polyploid because that gives you bigger fruits, bigger plants, and they're just more vigorous. This is showing you how way back when they actually did some, it's not really genetic engineering, I guess it is in a fundamental way, of the wheat plant. Wheat, the modern wheat that we use for making bread, amber waves of grain, is Triticum aestivum. That's the genus species name of your common bread wheat. Triticum aestivum, though, is actually a hybrid between 3 different types of triticum or wild wheat. So you have einkorn wheat, which is Triticum monococcum, Triticum siersi, and then Triticum turgidum. I'm not sure how to pronounce that one. And Triticum tauschii. So these plants, the hybrid between these 2 made this one, and then this one was then hybridized again. So you have multiple, the origin of these chromosomes is from multiple wild types of wheat. But hybridized together, we get the common bread wheat that we use, that we grow today. This slide is I just think it's a nice slide that summarizes the meaning of all the terms that pop up in this chapter, so I threw it up here. And that gets us to the end of chapter 6.